Recording in progress.
하나 둘 셋. 테스트 핀 마이크 테스트. 하나 둘 셋. 하나 둘 셋. 핀 마이크가 없대요. 핀 마이크. 하나 둘 셋. 내가 켜놨어. 내가 켰어. 하나 둘 셋. 테스트. 하나 둘 셋. 어때요? 핀 마이크 볼륨 좀 작아요? 핀 마이크 테스트 줍니다. 하나 둘 셋. 하나 둘 셋. 에이 하나 둘 셋. 하나 둘 셋, 하나 둘 셋, 테스트, 하나 둘 셋, 하나 둘 셋. 네.
Hey, Josh. You're muted.
I'm muted. Good to see you. Thanks for doing this. Looking forward
Absolutely.
to it. Hope all's well with you.
Things are well.
Good.
Good morning and good evening, ladies and gentlemen, depending on where you are at this moment. My name is Jury Wong, currently serving as the Head of International Affairs and Public Relations Division at Korea Biotechnology Industry Organization. It is my great honor to welcome everyone to the 2022 Global IR Pitching at JPM on behalf of 3 co-organizers, KoreaBIO, BioCentury, and Sidley Austin. This year's Global IR Pitching at JPM is our very second annual satellite event of the JP Morgan Healthcare Conference. This event is well known for its signature cross-IR pitching format where innovative biotechs and global top investors participate as panels. Through this event, we are hoping to see more cross-border licensing out deals and cross-border investments. We believe this global IR pitching event serves as a significant opportunity not only for the bio startup companies, but everyone in the entire ecosystem in bio industry. We would like to especially thank the Ministry of Trade, Industry, and Energy of the Republic of Korea for sponsoring the event. Global IR at JPM will be a 2-day program consisted of startup pitching sessions, VC panel discussion, and cross-border out-licensing deal case study sessions. And I assure you that you won't be able to see such a dynamic pitching event anywhere else. So stay tuned, and I hope you get the most out of the event. Today on our Day 1 event, I proudly introduce you to the 8 Korean biotech startups recruited by KoreaBIO, who will be pitching in front of many prominent prospective investors. For our investor panels, we have invited top biohealthcare VC PEs and IBs from Korea, China, US, Hong Kong, and Europe. And they are CBC Group, Life Capital, RM Global, Sofinova Investments, Tyborn Capital Management, KB Investment, KTB Network, Daily Partners, Intervest, and Partners Investment. But before we go straight into the company pitching session, we have invited 5 professionals who will talk about the recent cross-border collaboration trends, as well as some tips and hints about startup funding issues. I will now hand the session over to the session chair of the panel discussion, Jeffrey Cranmer, Executive Editor and Head of Daily Intelligence at BioCentury. Jeff.
Hey, thanks, Jerry. Thanks very much for having me moderate this panel. I am very excited to lead this discussion with these 4 guests who know far more than I ever possibly could about cross-border investment and deals. I'd very quickly like to introduce them, and then later in the panel, they'll be letting you know a little bit more about their firm's strategy, especially where it brings Korea into play. In the studio with you there in Korea, we have an old friend of mine from KTB Network, Ji-Woon Chung. He is Life Sciences Investment Director. And sitting next to him, we have Min-Sik Shin, Director of Life Science and Healthcare VC Investment Group at KB Investment. Joining us from New York City in Midtown Manhattan, we have Michael Keung. He is Managing Director and Head of North America for CBC Group. And hanging out in Hong Kong, just down the coast from where many of you are, Bo-Soon Hao, who's Managing Director and Co-Head of Private Investments at Tyborn Capital Management. Welcome, gentlemen. Very happy to have you on board. We are going to dive into cross-border investment in a little bit, but last week gave us a deal that's the perfect deal to start this discussion, as it's the sort of deal that I think every Korean startup would love to have in their portfolio. And I'm talking about ABL's deal with Sanofi last week. At 75 million, that's one of the largest upfront payments for a Korean biotech. This is a licensing deal for a Parkinson's therapy. And that deal, along with the now defunct deal between Bridge, another Korean biotech, and Boehringer out of Germany, I'm very curious, have Korean biotechs begun to come of age as potential partners in the eyes of Western pharmas? And do you expect to see more such deals in the near term? Ji-woon, I'd like to throw right to you and get your thoughts on that.
Yeah, Jeff, nice to see you again here. And also, talking about the Bridge Bio, you mentioned it. I used to be a lead investor of a board of director of Bridge Bio, and then I have felt that the more traction from the Western pharma recently. Actually, prior to ABL and Sanofi deal, there is another deal from KoreaBIO and Lundbeck. That was also a very big deal last year, and then I do believe that there would be more deals in the near terms because Korean biotech have tried to disclose their technology and data for several years in JPM or ASCO or in Bio USA. So, and the other reasons are ABL Bio and KoreaBIO have their own platform. So, which means that based on that platform, their second pipeline and third pipeline may have an opportunity to attract other partners in the world. And if so, their first deal would be a great reference for them. So I do think that there is more likelihood in the near term, and more deals are coming. I do believe that.
Excellent. Michael, do you have any thoughts on the deals that we're beginning to see come out of Korea?
Yeah, thank you. Thanks for having me, everyone. Good to be here. For sure, I think the Korean companies have done a lot of partnerships. And if we look back, maybe not just in recent years, but maybe 5-7 years ago, Hanmi Pharma did a string of big deals with Eli Lilly, Genentech, Sanofi, and others. I mean, we saw a big wave of partnerships that really put the Korean biotechs on the map. And it's great to see Sang-hoon, who's a good friend of mine at ABL, to do a nice partnership as well. And I think it's going to be inevitable that a lot of Korean biotechs will continue to do these cross-border deals, partly because a lot of the Korean biotechs are limited on capital to do these big clinical trials in the US. They do not, most of them don't have presence in the big markets of US and Europe, since they can't develop in those regions and don't have enough capital. Therefore, they have to partner out. So I think we will continue to see these cross-border deals. And of course, there's going to be validation of that platform, especially a company like ABL with this bispecific, showing that big pharma prove to them that they're developing these bispecific to the global standards. So I think it's a great sign to see. We hope that we'll see more. It's great to so Bridge Bio do the deal last year, and hopefully in the coming years we'll see a lot more as well. So it's an exciting time in Korean biotech.
Excellent. Thanks for those thoughts, Michael. You mentioned platforms, and obviously ABL has been developing their bispecific platform for a while. They have some ADC technology. Bo-soon, I'd like to bring you into the conversation now. Are more Korean companies developing cutting-edge platform technology that's attracting Western pharmas, Chinese pharmas? Is it platform tech, or are they developing first-in-class, best-in-class assets that now can compete on the global market? What is it that's drawing these MNCs in?
Yeah, I think generally, I mean, I think rational minds can have different views on this platform versus, assets. And I think the pendulum swings from time to time, but ultimately, if I put myself in the shoes of big pharma and big biotech, I think they're looking for really best-in-class, first-in-class assets. And that may derive from, very interesting platforms. And I think, Korea, in my mind, having been, in the industry now for 20 years or so, close to it, has always excelled, I think, in, very early kind of basic research, it's I think it has evolved, and I think translating that into the clinic, and I think that's where the relationships and partners, I think with the Sanofis and the large MNCs of the world can help accelerate and bring that to the global stage. But I think fundamentally, I don't just to comment here, I don't think innovation, no one has a monopoly on innovation, and I do think that with the introduction of as we've moved, and this is a very high level comment, but we've moved away from just small molecules here, like we're entering into new therapeutic modalities where I think it's a bit more of a level playing field. And so as you start to think about what becomes dominant over the next 5, 10, 15 years, cell therapies, gene editing, gene therapy, continuation of large molecules, I think you start to think about, look, innovation can be sourced globally, and we're not only seeing it here in APAC generally, but across the region in Asia Pacific. So I think over time the West will start to, I think, pick up and take more notice that you can find really interesting assets all across the globe, particularly in Korea and Asia more broadly.
Excellent. Thank you for that, Bo-Soon. I'd like to turn to Min-Sik and bring you into the conversation. Are there particular assets or technologies or therapeutic indications where you see Western and Chinese companies really looking to potential partners in Korea?
So thank you, Jeff. So there are many things going on in the Korean biotech ecosystems, and it's really hard to just pick one asset or technology. But I'd like to actually give a good example of this. Is that so recent? Within the last 2 or 3 years, one of the large pharmaceutical company called SK Biopharm actually have made lots of tech transfer, not only to the Europe, but also Japan and China recently. And as you can see in this case, they did a pretty good job actually selecting a niche market in the neuro area, CNS area. Lots of companies are working on the neurodegenerative disease, whereas this one picked up the anti-epilepsy medication. So that was another smart move there. And then the novel mechanism of action, the dual mechanism of action of the cenobamate, which was the anti-epilepsy medication that they licensed it out, that was also a good strategy. So these kind of assets that targets not only the major ecosystem, but also the niche market, that's being pretty much cooked in a lot of Korean private companies as well. So we'll expect to see a lot of those kind of assets in 5 to 10 years.
Excellent. Thanks for that. We did, I think it was Bo-Soon who mentioned that it's more of a level playing field right now, given the move away from small molecules, which I think is a really cool point. At the end of the day, though, I think, you look back to 2015 for when Korean biotech really kicked into gear. I think that was around the time that Hanmi started doing these deals. The funding began to flow a bit more. But that said, it feels, Korea's still fairly young on the global biotech scene in some ways. And I'm wondering, what are the biggest challenges that Korean biotechs are facing when pursuing cross-border deals? Ji-Woon, can we turn back to you for that?
Yes, Jeff, that is a great question. And then I can see a lot of changes when I jump into this industry from the pharma industry. It was like 10 years ago, but we don't have much actually. We didn't have much experiences at that time. So it's all about the experiences, such as the rigor issue and communication issues with media and then counterparty and so on. But right now, it's definitely not, because nowadays we know why we have to appoint kind of a Sidley for the global law firm to prepare the deal, because so our experiences has been accumulating. So currently, the bigger challenge is like about time and then the milestone itself. So which means that if there are more progress in the development, there would be a bigger deal. But considering the kind of a milestone resolution for the IPO or the other some fundraising or something that it seems that they need to have a deal at the earlier stage than they expected. So, but I do think that there is getting better because the investors also accumulated their own experiences that we need more time and that we can have a bigger deal. So that is almost at the end of educated period, I think.
Michael and Bo-Soon, looking at Korea from the outside, are there things that you perceive as maybe being hurdles or challenges that Korean companies are still looking for ways to solve?
Yeah, I'll take a crack at it first. I think it just historically there have been, I guess, deficient of venture capital funding for some of these companies when they were at preclinical stage. In particular, the CMC related work took some of the companies took shortcuts instead of completely validating the processes or optimizing. They'll get to the clinic first, only because that inflection point will drive the venture capital funding. And we've seen a number of times with a lot of the biotech companies that are in phase 1 or phase 2, a lot of the CMC work is still not the global standards. And we have seen some of the partnering deals fall apart because of CMC issues or assets returned as a result of it. So one thing that I would hope a lot of the Korean biotech companies spend more time doing is really optimizing the CMC early on. And hopefully the venture capital world also will be patient to allow them to do that, as opposed to trying to rush to get to the clinic where they think there's a value inflection point. I think if that could be done more prudently and more, I guess, robustly, I think a lot of these assets and the partnership will survive longer, and they could actually develop asset that could be a global standards. That's been so far our experience. We've done a lot of partnering with Korean companies, as you know, from CBC Group. As we have started companies, we partner with Genexen, we partner with Samsung, and we're doing cross-border deals in Korea as well. So we do see quite a bit of opportunities, but some of the deficiency we have seen, not the company we partner with per se, but generally speaking, has been on the CMC side.
Interesting point. Bo-Soon, does that ring true to you? Or are there other things that you've detected as you've done diligence on deals?
Yeah, no, look, I think Michael and CBC certainly have more experience, I think, specifically in Korea than we do, but I would echo those opinions. I think part of it is, I think, a lack of just, and I think this is improving, but the lack of not only dollars or resources, capital going into these early stage companies, but also I think the sophistication of the, and I think this is growing again, of the investor base there, really pushing and encouraging, things to be done at a global standard. I also feel that oftentimes there is a gap in the ability to fund these companies. So the aim is to get to, the public markets where there potentially could be a step up in value. But it may mean, again, I think, as Michael mentioned, shortcuts. And therefore, I think some of that has led to some, the challenges, I think, in consummating and taking forward, these collaborations. But I would say it's definitely improving. This is what I was speaking to previously. I think Korea has best-in-class, world-class kind of research, basic research that's going on. And it's really over time, I think, the development of that sophistication to take these assets going through the CMC work, thinking about all those aspects to actually get this into the clinic and ultimately commercialization. So I think it's just, it's a matter of time. It's a, somewhat of a nascent industry there, but the trajectory path to travel looks very, very good.
Excellent. Min-Sik, can we bring you into this?
Sure. So all three of you actually mentioned very important points. And I would like to just add two more points, which is more of the hands-on kind of elementary side of it, actually. So when I do the deals in the States, for example, and in Korea, there are some of the contrasts between those two areas. Number one is the way of communication, actually. So I like to always give an example of how the genetic was formed. Robert Swanson actually had talked with the professor in UCSF this is important, with a couple of beers. That's what they always say. And it's important saying that because it's always they're prepared to present their data with their storytelling in anywhere, wherever it's a casual talk. Whereas it's more, in Korean companies, it's more they need a table to get ready. So those kind of casual talk, it's driving what's the importance of the data. The second part is also the readiness of the data room, for example. So the CEOs or the management of the company in the US, for example, are busy actually making PowerPoints and Excels, telling the stories of their data into to the outside the world, and they're always prepared for the data rooms and they're always organized. And this is somewhere where Korean companies could actually take place in and then be prepared, and we see a lot of improvement there.
Excellent. Thank you very much. I want to turn now to investing, but really specifically to each of your firms, because it's not every day that we get 4 investors such as yourselves on the same Zoom, and I know those tuning in will be quite curious to learn a little bit more about your strategies and how you approach Korea. Michael, can I start with you?
Sure. So at CBC Group, we're about a 7-year-old firm, started back in 2014. We only invest in healthcare. We currently manage over $5 billion today. And we invest across buyout and private equity. We also do incubation, new company creations. We do have a venture capital fund, as well as royalty credit fund that's dedicated to Asia, and as well as a real estate play. We have 4 offices in Asia, as well as office here in New York. And we back definitely innovation and companies that could become leaders in their subsectors. As far as Korea is concerned, as I mentioned, we've done some partnerships where we look for really interesting assets. Just happened that we pick a lot of the biologics actually and monoclonal antibodies that we could bring to the Asian market. But now we also are going beyond just particular partnership, just say regional rights for Asia. We are looking for assets that could have a global rights. In addition, we are looking to make equity investments. And that style of investing, what we're starting to do is a more of a controlled buyout. You may have seen some press releases on some of the deals we're looking to do in Korea that can't really comment on, but we certainly are making a concerted effort to invest more in Korea as well. So we're certainly looking for deals, not only in biopharma, biotech, but across devices and diagnostics as well, since we're agnostic to healthcare sector.
Thanks, Michael. Bo-Soon?
Yeah, thanks. So Tyborn Capital, we've been going for about 10 years. We're kind of global growth equity investors, both in public and private markets. We have multi-billions under management. And I think our strategy with respect to healthcare, we invest both in public and private, sector agnostic, although I think we do tend to gravitate more towards, therapeutics. But, I think with respect to Korea, a lot of what we're doing, I think, with our investment strategy in healthcare is I think, similar, the topic here about cross-border. I mean, we do look for best-in-class, first-in-class, assets, platforms in their respective geographies. But then we start to intend to think about how, as we sit in Hong Kong and have reached both into the West and East, how do we help companies think about leveraging relationships, assets, access to capital, patients, navigate regulatory processes globally? So in certain instances, it can be simple as, helping these companies identify partners in respective countries, but, in certain cases as well, we'll actually start companies dedicated to bringing forth IP, let's say from the West into China, We're looking at other, other opportunities in the region to take, IP into other countries in APAC as well. But largely, look, a lot of what we do, I would say, 2/3 of it is crossover type investing. So, we're investing in companies that are 18, 24 months probably away from a liquidity event, meaning going to IPO. That's not necessarily where we look to sell, but that's actually where we think, because we have significant amounts of capital under management on the public side, we can scale up at the IPO, increase our exposure, and own these assets for many years. One of the panelists mentioned KoreaBIO. We were one of the largest international investors into that IPO. I guess that was 2020. Seems like ages ago, given everything. But and then we've recently invested in a Korean company in kind of the AI imaging space. So we're wanting to get more exposure into Korea. It's a place where we see a lot of opportunity. And again, I think these are businesses which I think ultimately we look for the ability for them to go global.
All right, let's go back to the main room. Ji Woon, could you tell us a bit about KTB?
Yes, thank you. So KTB is the first venture capital in Korea since 1981, and we are the first cross-border VC in Korea, and we opened the office in Bay Area in 1988, and also opened the China office as our first Korean VC at early 2000. And so cross-border deals for KTB is not a big thing actually, but in terms of life science and biosector investment, it was quite new for us when we think about back to the 2014 or something. But nowadays, and in Korea, since we are the first VC in Korea, so we actually try to do deal that like company formation deal from biosectors, from seed round and Series A round, and to the follow-on and follow-on. And then we want to be a very long-term partner for a Korean company. But when we see the cross-border deals, that strategically we try to jumping in at Series B and C round, and some give us other options or other options for the company, and then help them to the deal with the Korean company or Asian company or something. So so that is our strategy we have for the life science investment sector.
Excellent. And Min-Sik?
Sure. So KB Investment, which is 100% subsidiary of KB Financial Group, which is the biggest private bank in South Korea, has been established back in 1990s with current AUM, asset under management, of a little bit less than about $3 billion in currency. So we're one of the top investment firms in terms of the biotech area with the investment size. Also with a solid biotech team, from biotech industry, MDs and PharmDs and all that. So traditionally, which is also ongoing right now, we've been focusing on incubating biotechs in Korea and then giving their values, bumping up their values and create bigger values until the IPO. And even post IPO, we're trying to help them actually to put better Korean ecosystem. And a lot of examples, there are a lot of examples in the IPO markets. Right now, what we're been trying to do is that we're trying to be a liaison, or the bridge between the Korean ecosystem and also the global side. So example comes from the Arvel, for example, which licensed in the product of the SK Biopharm. We formed a company outside Korea, actually brought these products of Korean market into the global side, which was then acquired by one of the top pharmaceutical company in Italy. So we're trying to become a pioneer of the Korean pharmas, also a liaison that brings these valuable assets from global side into the Korean market. So that's our firm strategy.
Excellent. Let me stay with you. I think we have time for a final question, and I think it's quite an important one. Is what advice do you have for private Korean biotechs who are looking to add global investors to their syndicates, What tips do you have for Korean biotechs who are looking to get in front of not only global investors, but also people such as yourself?
So there are so many things that we could answer, but to make long story short, when we think about, outside Korean investors actually putting capital into Korean company, for example, they want to be also, I'm sure everybody here knows that they want to be not only the one-shot investors, but they actually rather want to be a lifelong partner. So rather than having Series B or Series C just one-shot investors, they like to give input of their thoughts from the seed round probably, or at least in a sequential way. So I think that's the motto that a lot of not only Korean investors but also the global investors have, and this is what something is to be keep in mind and have a good relationship based on this kind of idea.
Jiwoon, how would you answer that?
Yeah, so since time is out, and then I'm trying to give a very short advice, and I do think it's all about the language actually, and then which means not only for the communication, but when the Korean company to prepare their own the conference information, they have to prepare their own the English material as well at the same time, and then because when global investor is doing the due diligence, they need their history, but they should be based on the language. Not only in Korea but also in English. So I do advise the Korean companies to prepare about your material at the very early stage in English at the same time.
Yeah, that's from where I sit at BioCentury. I am going through 500 to 1,000 pieces of news a day. A lot of new companies, a lot of very well-funded new companies, and given that flow coming from all of this innovation, you need to make it easy for people like myself to digest the information quickly. I'm working on my Korean. I've got mashita down. I'm told that that's the only word you really need to know in Korean, and I'm going to go with that. Michael, tell me a little bit about what your advice would be.
Yeah, I think it would definitely be persistent. I think continue to reach out to the funds and give updates as often as you can. And certainly when you do that, I know the scientists are very smart and they could do really well, but don't oversell and underdeliver, but perhaps overdeliver, right, by underselling. But I think that persistence is going to be really important. So we look forward to meeting a lot of companies. Since we don't get a lot of deal flow passively, we actually have to call them. So we hope that a lot of companies will continue to reach out to us.
Excellent. And then a last thought from you, Bosun. I know that you mentioned you're more of a crossover, so companies might be coming to pitch you a little bit later in their cycle. But what advice might you have?
Yeah, I mean, look, we like to get to know companies as early as possible, just because we can track the process or track kind of progress, and ultimately we're betting on people. So I think it is important to establish that relationship. I mean, I do think look, English materials are critically important, but ultimately I think it's in biotech, it's the quality of the data and how it's been generated, and is it being generated in global standards. So I just implore, and that's probably the universal language, right? So I think ensuring that you're just taking a global approach to the process, your processes, and generating the data that then can just, that doesn't matter, doesn't care what the borders are. I just think that's critically important. I also think, look, a lot of the collaborations that happen across academia at the kind of earliest levels, I think there is collaboration between West and East, and I think leveraging some of those relationships that you have advisors. I often see a lot of US and Western advisors with Korean companies, and I think that could also be another way to get access or in front or mind share in front of US or Western investors.
Excellent. Well, thank you, gentlemen. We could go on for another hour, but unfortunately that's all the time that we have. There's a great program ahead, so I'm going to turn it back to Jury, and thanks for having us on for this chat.
having us.
Thank you.
All right. Thank you, Jeff. I guess we transition.
Yeah.
Well, thank you, Jeff, and the four panelists, Michael, Bosan, Jiyoung, and Min-sik. I'm sure this vital information was helpful for the startups who are eager to see cross-border deals. Now we will move on to our highlight session of our day, which is the 15-minute startup pitching and investor Q&A session. Joshua Berlin, Executive Director and Editor of New Venture at BioCentury, will be our moderator for this session. I'm thrilled to see this session. Here's the mic, Josh.
Thank you, Jury. And just on behalf of BioCentury, I did want to thank everyone again for joining today's Korea Biotech track. You know, BioCentury has followed the emergence of the Korea biotech sector now for several years. This includes a popular Korea Spotlight series that's written by Jeff, who just moderated the earlier panel. And it also includes a Korea Biotech track that we host each year in November at our China Healthcare Summit. So it's a real honor today to join with KoreaBIO and Sidley Austin for the second year on this program. And I do think KoreaBIO has assembled a great lineup today of presenting companies from Korea. And then we also have a great lineup of investors from Korea, Asia, and the West who will provide feedback and insight to these companies. The VCs will ask questions to each company following their 10-minute presentation. So let's get started with our first presenting company. Our first company today is Abtis. Abtis is developing a pipeline of antibody drug conjugates based on its site-specific linker technology. And presenting for Abtis is Chief Business Officer Justin Oh. Justin?
Hello. Thank you for the invitation for today's session, and we are glad to make our presentation for Abtis. Abtis is an ADC development company, and it's glad to hear that about the platform technology by the previous session. And hello, Michael. Good to see you again. We were founded in 2016 by Dr. Chang. He's a professor at Sungkyunkwan University at the School of Pharmacy. And we have been developing ADC therapeutics development. And we have 20 employees, but a few more people will join us next month. And we are located in Suwon and also in Seoul. We were created in 2016 at Korea Seoul BioHub. And between 2021, we got the Series B funding. We have been developing our linker technology, AppClick standard and AppClick Pro. And we were selected in 2020 as one of the baby unicorn company. And these are our current investors, Stonebridge, K2, JX, CKD, Timefolio, Prodigy, Azure IB, KDB Capital, and Kingo. So what kind of technologies do we have? We do have site-specific conjugation. We are ADC linker platform technology company, and our technology has the site-specific conjugation. And distinguished point about our linker technology is that no mutation of our antibody are required, and stable and cleavable linkers. So these are our top management, and Dr. Chang and the CEO, and he has been developing ADC linker technology for more than 10 years, even before founding his company, Abtis, 5 years ago. And Dr. Oh, and he used to be also CTO of LegoChem, another well-known Korean ADC company. And he has been the CTO of our company since last year. And Dr. Choi graduated from Yonsei University Medical School with an MD and PhD, and he has been developing experience and clinical experience for more than 20 years. These are our SAB members. And what is the business model? I mean, we are an open innovation company, so we do have partners in, for example, in the antibody development companies and also payload small molecule companies. So what is our competitiveness of our technologies that, as I mentioned earlier, drug conjugation in not engineered monoclonal antibodies, and a site-specific conjugation, and the payloads can be flexible too, and stable in serum, but also cleavable within the cancer cells. So these are how the ADC conjugation on the antibodies actually happens. It's a principle for site-specific antibody conjugation by using our technology. And this is a simple two-step process of the binding of our linkers to the Fc part and the specific lysine on the Fc part of an antibody. These are the major two technologies of our company. And when you see the AppClick Pro in the bottom, there is a simple two-step process to make the conjugation. The first part is the antibody linker conjugation and the drug linker conjugation. And we are targeting the solid cancers using this AppClick Pro technology. As you can see on the physical chemical properties of our using our technologies, that you see the simple clean three peaks when you look at the mass spectrometry, and this means that we have a clean dot 2 of a drug-antibody ratio. That means that two drugs will be conjugated on the antibody. So what's the advantage of using our ADC linker technology is that obviously you don't have to mutate the antibodies part, so it's easier on the CMC part, and also it's a simple two-process, so it will save some time, and we are improving on our yield, so that we expect there will be many advantages using our technology. These are the example HER2 ADC, and we will show you. This is DAR2, drug-antibody ratio 4, using our linker technology and using the trastuzumab antibody. This is the in vitro cytotoxicity using our comparing actually our HER2 ADC to Kesala, which was developed and marketed by Genentech and Roche. The red line is our HER2 ADC, and the gray one is a Kesala. And comparing this the targeting, we are doing the in vitro cytotoxicity. Ours has a much better efficacy and shows IC50 value of a single-digit picomolar in HER2 positive cells. These are in vivo efficacy. The NCI and 87 CDX model, and minimum efficacy dose of our HER2 ADC is less than 3 milligrams per kilogram. As you can see on the tumor volume and the pictures of the tumors, you can see our HER2 ADC has been very pronounced differences effectiveness on the CDX model. This is a GYM2-1 CDX model. This is especially good data because GYM2-1 is Kesala resistant cells. So our ADC, HER2 ADC, has shown strong result compared to Kesala. This is a multiple. Basically, with multiple dose, I mean, there's nothing there, as you can see in the pictures. Human plasma stability is very comparable to the actually the trastuzumab monoclonal antibody itself. And pharmacokinetic studies and Cmax of the free MMAE using our ADC, HER2 ADC is very small, and also the ratio is very small. So it's very clear. It proves that our linker is very stable in the healthy cells. So I mean, we are ADC linker platform technology, also developing our own pipelines. We're using our technology, and it can be the technologies can be very versatile. You can put the radioisotopes too. We are doing a collaboration with the Seoul National Medical School using on the radioisotope also at this time. This is a collaboration model that we have partners with other payload small molecule companies and the antibody development companies to develop ADC together. These are the pipelines. The major pipelines are HER2 ADC. We are getting ready to into the preclinical stages, and also Claudin 18.2 ADC. We are also ready to move into the preclinical stage this year. And the Claudin 18.2 antibody itself, we have licensed in from GenScript recently. And thank you very much for hearing the presentation. Any questions?
Great. Thank you so much for the presentation, Justin. Michael from Seabridge or CBC Group now, maybe you could take the first lead on the questions.
Sure. Justin, great to see you again. Congratulations on your move. Good to see that you're part of the exciting company. We certainly love the ADC space as we are bringing Trodelvy from Gilead and Immunomedic to South Korea through Everest Medicine. And we do believe ADC is a great way to create platform companies as well as create different drug candidates. So maybe can you just speak a little bit in terms of your IP? Obviously, you have this interesting toxin, I guess a payload with MMAE that you have in your HER2 and others. Or maybe just give us an update on where you are in your IP, especially on your linker technology.
I mean, we already filed the international patent filing, and we are in the process of filing individual countries. For example, AppClick standard, we filed, and we got the approval from the Korean Patent Office that our patent has been filed and approved by the Korean Patent Office, and we are in the process of filing to each major countries, like in China, Japan, and also the US and Europe. That is the stage at this time.
And I guess in terms of your linker technology itself, how does it differentiate from other guys out there, and especially with Gatesella and others? What would the key differentiation, I guess, is it in the improve in the safety profile? You think is the cell penetration? How would you differentiate?
I mean, the technology-wise, I mean, the biggest difference is the site-specific conjugation, and also you don't have to engineer anything on the existing antibodies. So that is the technology point of view. That's the biggest difference. And the actual, I mean, as you can see, the PK data also. I mean, because of the no engineering, no mutation required on the antibody themselves. So we think the existing characteristics of an antibody will be maintained even after the conjugation we're using our technology. So that, I mean, we need to definitely do more data. We need more data. We were planning to do the monkey tox data within this year. So we think that immunogenicity, for example, using our technology can be better data at the end of the day. But I mean, we have to show to you guys about the actual data too. So we have some way to go, but this year, I mean, we are expecting to get the good data using the monkeys.
Thank you.
Great. Thank you so much, Justin. Really enjoyed your presentation, and we look forward to hearing more.
Thank you for your presentation. I have a question about your HER2 targeting ADC product, and you have compared your product with the Gatesella, but as you know, there is second generation HER2 targeting ADC came out already. So,
in
HER2, yes. So do you have any evidence for your product will be better than second generation ADC product?
That's a good question. Actually, I am receiving that question quite often these days. And actually, we are planning to do the comparison test with the Daiichi Sankyo SAR2 ADC, and we actually made a request to the US CDMO company to produce the INOTU2 in addition to ours. So we will do the comparison test very soon. I mean, INOTU is great. I mean, their ORR is quite amazing. I mean, that's why people are expecting that it will be a blockbuster drug in a short time. But at the same time, they have a tox issues, especially in the lung side. So they have a black label on their drugs. So we think we do have a niche market in that area too. Also, I mean, last week, KoreaBIO announced the HER2 ADC deal, more than $1 billion with Ixuda. So I think there is some room. So it's a competitive landscape, but we think we do have some room there too. But our also another pipeline, Cloud 18.2, I think it will be our one of the major pipelines too. Thank you.
Okay, great. Thank you so much, Jason. I think we're just about out of time, but I'm sure if there are other questions, I'm sure our investors would be happy to follow up with you directly. So thank you so much for the presentation.
Okay, thank you.
Yep. We are now ready for our second company. So the next presenting company is KoreaBIO. KoreaBIO is engineering oncolytic adenovirus to deliver RNAi therapies, simultaneously targeting 2 cancer signal pathways. Presenting for KoreaBIO is Strategic Development Lead, Andrea McDowell. Andrea?
Hi, it's very nice to be here today, and I just want to say thank you so much for giving us the opportunity to present our platform technology as well as one of our lead assets that we have in development at this stage. And so thank you also for the introduction. So I am Andrea McDowell, the Strategic Development Lead at KoreaBIO. And so we're just going to go and get started straight ahead. So briefly, I would like to introduce to you KoreaBIO's core and main mission that we are using our bispecific shRNA technology in order to address. And so first of all, I'd like to say that the main reason that we are using this technology to target these critical unmet needs is that we're aiming to address the core gap that is existing between basic science as well as the medicine. And so what we see oftentimes is that while there are a variety of different targets that are identified in the human body, there's a big chunk of them that are actually undruggable. And this is through the fact that they're unable to be easily accessed through different types of conventional medicines that are existing at this time. And so our kind of strategy is, of course, we want to be able to act at the genetic level instead of at the protein level. And so in our perspective, we think that RNAi technology has a really, really great potential to be able to overcome this core critical unmet need. And so some of the specific ways that we are looking to overcome this, specifically in the case of immune checkpoint inhibitors, which, as you all know, is a very, very exciting advancement in the field of oncology. However, we can also see that there's a tremendous need to be able to overcome the relatively low response rate that is seen in the patient population. And then also we want to be able to address issues in tumor heterogeneity as well as in the development speed and these kind of issues that we typically see in drug development space. And so to give a very brief introduction into KoreaBIO's background, so you get a little bit of a better idea of who we are as a company. So we were founded in South Korea in 2016, and we had our pre-A financing round in 2018, followed up by Series A, as well as a bridge funding round recently last year. And here I just want to emphasize in our corporate structure, I think one of the really main strengths that we have as a bioventure company is that we have a very, very strong and robust in-house R&D capability. And this is seen in both the production level, as well as for IND-enabling studies in vitro and in vivo, as well as our bioinformatics team. And then also just to give a bit of more of a face to who our team is and who we're being led by. So we have both our internal management team as well as an external scientific advisory board that's continuing to grow. And these are some of the forefront experts in both the cell and gene therapy space, as well as specifically as we are the pre-IND stage at this time, we're looking towards people that are experts in bringing things from the bench into the clinic and then through commercialization. So we have a very, very strong team that's leading us with experts both domestically in Korea as well as internationally abroad. And so very briefly to give the introduction to our platform technology. So especially at this phase, we're focusing on our application of a bispecific shRNA, and especially in the space of oncology. And at this time, we are putting into the oncolytic virus, specifically adenovirus 5.3, but we are also looking towards expanding into different types of delivery systems so that we can target other indications, as we can target any 2 different types of disease-related genes. So at this time, just to give a brief background, for RNAi component, our core strengths is that, as I mentioned, this is a bispecific shRNA. So this is our very, very unique component of our technology and RNAi platform, and the fact that in a single shRNA, we are able to target 2 different oncogenes in the case of oncolytic space. So for example, mTOR and STAT3, these are both very, very desirable targets that we are able to have the synergistic effect, which is very critical also in the cancer space, as one single target knockdown is able to be easily avoided and overcome in cancer. And then also in terms of the safety side, we can see that by having no passenger RNA, as both of the strands of the shRNA are able to have the efficacy effect. So we can also see a reduction in the risk of off-target effect, as well as by more stable production by a single promoter. And briefly for the oncolytic virus side, we also specifically selected the oncolytic adenovirus with the tumor-specific targeting capability, as well as very importantly from the safety element for the tumor-specific lysis, and the fact that the virus is only replicated in the tumor cells and the cancer cells that have these cancer-specific transcription factors. And we're also having ongoing efforts to be able to enable systemic administration. And so to give a very brief background into what the process that goes into for the design of our bispecific shRNA, what we can do for the case, just as an example, for our lead asset for mTOR and STAT3, we find the reverse complementary siRNA producing strands, and then we do some screening processes, and we can find the most efficacious candidate in knocking down the mRNA of the respective genes. And then as you can see here also at the bottom panel, we can actually demonstrate that when comparing to a dual promoter approach, when we're using 2 different shRNAs to target and knock down 2 different genes, we can actually see that we have a much more even knockdown at the total cell level, as well on the bottom right panel, the single cell level. We get a nice little cluster at the bottom in the blue, using our bispecific shRNA. And then also very briefly, I would like to mention that in the RNA therapeutic competitive landscape, we can see that there are a great amount of trials that are going on, improving the validity of the RNAi system. However, we can see that based off of the structure commonly used GalNAc in this space, unfortunately, there's an issue with what is the indications that realistically can be targeted. And so this is one of the ways that we feel like, particularly starting off, we want to really be able to penetrate into this not really existing oncology-related RNAi capability. As you can see here, most of the indications are mostly related to liver, and this is due to the tropism that's associated with this RNA structure. And then here briefly, we want to be able to give also a bit of a background into this mTOR and STAT3 pathway that we're targeting in our lead asset CA102, which is currently in the preclinical phase, but we're anticipating to get into the clinic sometime next year in Korea. And so just to give briefly this rationale, we can see that these targets are very advantageous because currently mTOR and STAT3 don't really have very many effective or safe therapeutics available on the market, and we can see that they have the synergistic pathway to have a very nice synergistic effect when they're both knocked down. And so we're targeting primarily bladder cancer as well as head and neck and cutaneous squamous cell carcinoma. And so looking here also, we can verify before going into doing any type of preclinical development or clinical development. And we can see here that by evaluating in silico level the clinical data of, for example, bladder cancer patient, we can see that when mTOR and STAT3 levels are very, very highly overexpressed in bladder cancer patient, there can be a very dramatic and significant reduction in overall survival over time. That is not observed in only the overexpression of mTOR or STAT3. So this is another way that we're validating our target selection. And then further, so just to reiterate, we're incorporating our mTOR STAT3 bispecific shRNA into oncolytic virus currently at this stage. And so we can see there's a very interesting effect in the dramatic increase in the amount of the siRNA produced by CA102 compared to CA10G. And CA10G is just the oncolytic virus itself for your reference. And we can see that there's a nice relative knockdown expression for at both the mRNA and the protein level. And this is reiterated in the single cell transcriptome profile between CA10G and CA102 treated bladder cancer cells. And we can see that the gene expression is knocked down in almost all of the cells that were treated. And then also we can note here that looking at the anti-tumor efficacy in both bladder and head and neck cancers that are pre-treated with CA10G compared to CA102, we can have almost complete elimination of the tumor. And then this was further validated in a combination therapy study that included cisplatin as well as our CA102. And we could see at the bottom purple line here, there was a very, very nice reduction in additive effect when we had the combination of CA102 and cisplatin. And so just in conclusion, I'd just like to give the overall big picture of our advanced drug development strategy. And so as I mentioned, currently at this time, we're looking towards the viral system implications, but we're also working on development for also non-viral system in order to expand our indications beyond cancer and to see what this platform is truly capable of. So these are, we're looking to expand for our delivery system as well as other combination therapy. So thank you very much for your time, and I'm open to questions now.
Great, thank you so much for the presentation, Andrea. Why
Thank
don't
you.
I turn it over to Bosun Howe of Tyburn Capital Management to lead the Q&A to start with. Bosun?
Great. Yeah, Andrea, thanks for the presentation. Really interesting platform you have there. I guess the first question is really around your bispecific. I mean, it's that's quite kind of novel. When you think about that approach with a single promoter, how do you manage kind of the ratio of just kind of expression right between the two oncogenes? Also, how do you think about typically one of the challenges around shRNA anyway is oversaturation. So like how do you, with the RSIC, like how are you guys thinking about that? And have you seen or managed or seen any issues related to that?
So something that's actually really interesting in that respect is that if we're looking at not only the issue for, as you just mentioned, the risk entry issue, right? Which of the RNAi inducing silencing complex is going to be attaching to which strand and being produced in which way. And so something that's actually really interesting that we've seen is that in our case, I think you probably saw in our slide earlier that there was a differential expression for the siRNA for the two targets. But what's really interesting is that in the actual the cancer cell of interest, we could actually see that there's even the differential expression of the mRNA for STAT3 and mTOR. So it's actually something that we can target to actually have a benefit and an advantage to be able to have a little bit higher expression of the siRNA for the STAT3 compared to the where our mTOR. But then we actually see in the cellular level, there actually is the matching overexpression compared for the STAT3 over the mTOR. So we can actually see this type as an advantage in the production. And we can also select based off of which of the different combination of our sequences that are going to be able to match the cellular profile in terms of the ratio of the siRNA that is produced by our shRNA sequence. So thank you. But I think also the key point is that this is already something that has to be factored in, but this is something that we can eliminate the compounding issue by having the differential expression that's also contributed by a dual promoter system. So I think that this is a way that we can really have a more deeper control. So thank you very much for that question.
Just another follow-up question. I know you're using Ad5-3 for the vector. I mean, can you just talk about why you think that has particular advantages, and then have you seen any immunogenicity or immune kind of response issues related to using that?
Mm-hmm. So we specifically selected adenovirus 5.3 in this serotype because we particularly felt that it combined the particular safety profile advantages of the adenovirus as well as by modifying the knob so that it's able to have a bonding with more of the different types of the protein receptors that are more commonly expressed on the cells. That's not seen in the other serotypes. So it's kind of having the best of both worlds. That's kind of the way that we saw in terms of the higher binding efficacy with our cancer cells and targets of interest, as well with the safety profile that could be, a potential issue. But in any of the in vitro tests that we've conducted and in vivo tests at this time, we didn't really see any issues with immunogenicity, but of course this is something that we are going to be verifying further before we're going into our phase 1 study.
Really last question I have before kind of allowing others is really on your lead program. I think you're going after BCG resistant bladder cancer.
Correct.
This is more of a competitive landscape question. I mean, we've seen some of the IL-15 therapies demonstrate pretty interesting efficacy there. So as you kind of go potentially head to head against some of these programs, how are you thinking about the competitive landscape? I mean, bladder cancer lack of innovation. Probably BCG has been the standard for the longest time.
Right.
So there's clearly room for innovation, but we have seen, I think, increasingly more competitive entrance into that area.
Sure, and that's a really great point as well. And so I would like to point out bladder cancer isn't our only indication of interest. We're also looking into several other options at this time moving into the clinic, including head and neck as well as cutaneous. And also we're looking into pancreatic cancer recently as well. And in the case of bladder cancer, as you mentioned, it is a very tricky target and a tricky market to go into. But we really feel that by specifying the BCG resistance, I think there's a really, really tremendous unmet need. And while there may be other types of therapies that are coming out at this time that could be able to address these issues, I think really the advantage of using this type of platform is the fact that we have the dual effect of the oncolytic virus that is able to induce apoptosis in the cells as it's producing. And then we're having that kind of further additive benefit by the shRNA that's knocking down the mTOR and the STAT3. So we think that one of the key advantages could be the higher efficacy of this dual system approach and also the safety profile that's really nicely associated that could not be seen particularly in some other different types of therapeutics. So
much.
Yes, thank you.
Great. Thank you so much, Andrea. Really, really appreciate it. We're about out of time, but thanks so much for the presentation, and Bosun, thanks for the questions.
Yeah, thank you so much.
So we're now ready for our next presenting company, our third company. It's a company called ENG, which is a cell and gene therapy CDMO. Dr. Yoon Hee Kim, Deputy Manager of Business Development, will present for ENG. Dr. Kim?
Hello, I'm Yoon Hee Kim from ENG. I'm very glad to be here to introduce what we do in ENG. I'm sorry. Okay, let me start. I apologize. ENG was founded in 2018 March as a second spin-up from Samsung Medical Center. Our headquarters office in Cheongdam-dong in Seoul, and our research institute and GMP facilities in Hanam City. And our business focuses on cell and gene therapy, CMO and CDMO service. And we are also developing our own pipelines. And so far, we raised about 30 million in USD for investment successfully. And I believe we are the only biotech startup company that Samsung Group has invested in Korea. Our CEO, Dr. Jong-Wook Jang, is currently a professor at Samsung Medical Center. He's the head of GMP operation at Cell and Gene Therapy Research Center in the hospital. And he's the former director of R&D division in Medipost. It's quite well-known Korean stem cell therapy company. And he dedicated his career to develop cell therapy and GMP establishment and operation, and still very active on academic side and doing a lot of consulting activity for government and industry. Besides our CEO, we have very strong science and technology background, including our management team. More than 30% of our personnel has master or a PhD degree, and we are very organized for R&D division and also cell and gene therapy manufacturing division. So let me talk about our business area. We are providing cell and gene therapy, CMO and CDMO service for R&D pipelines to reach to market. The GMP and regulatory affairs, regulatory science issues are the bottleneck for the commercialization. That's where our CMO and CDMO service comes in. So we have very well-established quality management system from production and QC and QA. and packaging, and also with equipment and material management. And we also running very stringent documentation system. So let me introduce you our first GMP plants. We have it's in Samsung Medical Center. We have 4 clean rooms and 2 pre-sterilization rooms and QC lab and storage. Here we are manufacturing clinical grade cell therapy products on a campaign production basis with a very stringent monitoring system and also decontamination system and also independent HVAC system to prevent the cross contamination. So since 2020 August, the Advanced Regenerative Bio Act got effective in Korea. So it's mandatory to have human cell management business license to run cell CMO business in Korea. And plus we have got ISO 9001 certificate for our quality management system. Since we launched our business in 2018, we have been performing 19 projects with 14 different clients, and we've got 5 IND approvals from KFDA so far. So we know how to handle the regulatory affairs in Korea. And the products we manufactured were safely administered to 28 patients so far. So we are actually leading the cell CMO market in Korea. So if you are interested in Korean market for cell and gene therapy, please contact us. And our annual revenue is fastly growing. Until last year, we achieved $5.9 million USD. And for next year, we are aiming to have another 8.6 million. So we analyzed the CMO project we have been doing, and we noticed the number of cell-based immuno-oncology project is increasing. And as we expand our business to meet up to the increasing demands for manufacturing CAR immune cell products, we constructed second GMP plants. It has negative air pressure room, so we can deal with the virus work. And the construction was completed last year, September, and we acquired human cell management business license last month for this facility. And here we can manufacture the products like CAR-T and CAR-NK. These are the photo of our second GMP plants, and it's process development room and negative air pressure room and positive air pressure room and our QC labs and storage facility for raw material and DS and DP. So now we are constructing third GMP plants for virus production, and it will be ready for the production by June 2022, this year. And here we are going to manufacture viral factors such as LANG-T and AAV, and also clinical grade gene therapy products. So now we have 3 GMP plants. First one is for cell therapy products, and second one is for gene-modified cell therapy products using virus like CAR-T and CAR-NK. And third one is for virus vector production such as LANG-T and AAV. So this means we can provide one-stop service for gene-modified cell therapy products like CAR-T. So for clients, it's not really easy to make independent contracts for each process like DS processing and plasmid DNA production and virus production and CAR-T cell expansion. With EN cell, it's possible for a client, they need to make only one contract for one-stop service. So now, let me move on to our pipeline. We are developing next-generation stem cell therapy for rare muscle diseases like Charcot-Marie-Tooth disease and Duchenne muscular dystrophy. So it's from umbilical cord drive MSC, and the treatment effects comes from the paracrine effect of stem cells. And EN cell technology increases the cell yield in the initial part of the cell manufacturing process. So we can harvest the drug product only in passage 3, which is quite early compared to other stem cell therapy products. And also it's in cryopreservation form, so it could give us a lot of advantages for logistics. And we applied for the patents for our technologies for cell isolation and adaptation and screening. And EN cell technology could reduce a whole lot of time for manufacturing the cells and could save the cost. And the drug products are much younger and fresher, so it shows improved treatment efficacy. And like I mentioned, the cryopreservation technology could give us a lot of advantage for logistics. So we found that XL1 is very important cytokine for inhibiting apoptosis in muscle tissues. So it could be applied in muscle disease treatment. So we named it as EN001 and continue to develop it for Charcot-Marie-Tooth disease and Duchenne muscular dystrophy treatment. So we finished POC studies and also completed the GLP toxicity studies for both indications. And okay, so Charcot-Marie-Tooth disease is a rare muscular disease. It's caused by demyelination in Schwann cells, and it leads to the loss of sensation and motor control in the patient's limbs. So the hands and feet will get deformed eventually. And currently, there's no really effective medication in the market. So this is our POC data. We observed the EN001 recover the myelin sheath in the nerve system. And we also observed the nerve conduction velocity recovery and also in motor coordination recovery. And this is the Rota-Rot test we performed. And the CMT mouse without any treatment lasted on the treadmill for 8 seconds. And the mouse number 1 was administered with 5,000 cells of EN cell. It lasted for 51 seconds. And the mouse number 2, it was administered 50,000 of cells and lasted 93 seconds. And the number 3 was administered 500,000 cells and lasted 109 seconds. So it works in dose-dependent manner. So just to save the time, I'm just going to move on to the next slide. So we actually performed a repeated administration and we confirmed the same result, but it's still unpublished data, so it will be disclosed later on. So we found that fibrillin 5 is important cytokine for regenerating the myelin sheath, so it got published as a featured article in Stem Cell 2020. And Duchenne muscular dystrophy is also a rare muscle disease. It's a maternally inherited disease, so it affects mostly little boys and girls. And they have some mutations in dystrophin. And most of the patients die before they reach to the age of 25, mostly heart attack, because their heart muscle will fail at some point. And actually, there are 2 gene therapy for this disease. But they cover only a small portion of the whole patient population because they're working in a limited mutation range and type. So there's really no effective cure in the market. And this is our POC data. And EN001 shows anti-fibrotic effect in the muscle in DMD mouse. And we also observed the myogenesis marker, MHC, was upregulated. And while the annexin 5, the apoptosis marker, was downregulated. And also we observed the serum creatine kinase level was significantly downregulated. serum creatine kinase is a very important blood biochemistry marker for DMD patients. Normally, it's very high in the patients. And we also observe the increased motor ability. And also we...
Yunhee, we're just about out of time. I wanted to make sure we did leave enough time for a couple of questions, if that's okay.
I'm sorry, it's almost to the end. Could I just spend just one more minute? Okay.
Sure,
so,
one more minute, thanks.
Okay, well, I apologize. So we've got the approval for phase 1 clinical trial for both indications, and it's ongoing. And actually, the first administration for the DMD patient was performed successfully like 2 days ago. And we are aiming to get the conditional approval for both indications by 2025. And also, we are preparing for IND filing for US FDA to go to global market. Thank you for your attention. I apologize for the delay.
Thank you so much for the presentation. Let me hand it over to Dr. Noel G. of Sofanova Investments to perhaps ask a question, Noel.
Maybe starting with your CDMO business, how large do you expect the Korean market to be, and what are your expansion plans to other regions in terms of business and providing cells and gene therapy for other areas? And will you be Asia limited, or are you thinking Western countries as well?
Well, actually, it's been only 3 years since we start our business. Now we are expanding, but eventually we have to go to the global market, and it should be not just for Asia or Asian Pacific. It should be the whole world, maybe. But at the moment, we are considering Asian Pacific market initially. That's actually the real possibility at the moment.
And if you don't mind, a follow-up question there. Thinking about moving forward, you have your company has quite large ambitions for a young biotech running a CDMO business on top of going through development of your own asset. How are you considering resourcing the drug development piece versus the CDMO piece moving forward? And where do you imagine most of your growth will come from in the next 5 years?
Right. Actually, it's not really first time we've been asked that question. We've been asked that question, the same question, quite some times, because we are aware that there could be some conflicts of interest issues, because we are developing our own cell therapy pipelines and we are doing the CDMO service for other companies. So for future business, we are considering, actually, we are discussing about the spinning off one of the business separately. So the customer wouldn't be so worried about their technology. So actually, eventually it should happen, and we are considering it. Nothing is definite yet, but we are aware that issues.
Okay, great. Thank you so much for the presentation and for the questions, Noel. I'm afraid we're out of time, so we're going to move on to the next company, but thank you so much for your time today. So our next presenting company is a company called Genealogy. Genealogy is using computational tools and DNA testing to identify cancer risk. Presenting for Genealogy is Chief Technology Officer, Dr. Boom Han. Dr. Han?
Oh, yeah. Thank you. Okay, today I'm going to introduce our new technology called the fast RNA. I'm seeing my face there, so. Slide, please. Well, I'm in my slide. Technical. Should I stand there or? Introduce myself. Okay, I'm a professor in Seoul National University. I'm the best university in Korea. And I did my PhD in computer science. I developed computational algorithms and methods for audio analysis, genetic analysis, biological analysis. And what should I say? I work with our CEO, Andy, and should I just, so you guys are, oh, thank you so much. Okay, let me go on. So our technology is an AI-powered single-cell RNA analysis solution that is 150 times faster than the current solutions and 50 times lighter, requiring much, much less computational resources, and is fully interactive. So why are we looking at RNA? RNA represents dynamic changes of DNA expression and has rich information that can sensitively detect an individual's current health state. So the interest in RNA has been steeply increasing in both academia and pharma. Traditionally, people have been using bulk RNA. Given a tissue or an organ of interest, bulk RNA gets average RNA information from all types of cell types. Therefore, the interpretation is at most fuzzy. Thanks to the recent technological advances, we can now get the RNA information from each single individual cells. With this information, we can have much more clearer interpretation. The market for single-cell analysis has been increasing. In the past 5 years, the single-cell RNA analysis market grew from 0.7 billion to 3 billion. 40% growth rate has been reported for NIH grants from 2018 to 2021. Clinical applications are wide, including health status monitoring, disease diagnosis, and personalized drug decisions. Because of these wide applications, we expect our market to grow to 6 billion in the next 5 years. One interesting and very critical problem of single-cell RNA data is the characteristic called what we call unknownness. When the researcher gets single-cell RNA data from the machine and NGS machine, within the data, there are no cell type labels. So cell types of the cells are completely unknown. We do not know which cells are CD4-positive cells or monocytes. This prohibits a direct and intuitive interpretation of the data by humans. Bioinformatics, BI downstream analysis, therefore, is a must in order to correctly identify the cell types of the cells and to interpret the patterns in each cell type. Therefore, the role of BI is much greater for single-cell RNA data than for other technologies. Yeah, but the bioinformatics analysis is really in chaos now for single-cell RNA. There are about 10 different steps for single-cell RNA analysis, and for each step, there are so many choices and methods and algorithms are coming out and too much parameters to optimize. So there's no really single gold standard that fits to all data. So researchers are so complicated, and they sometimes pay for commercial services, but the commercial services are very simplistic and do not allow tuning or optimization. Some researchers, therefore, try to build their own pipeline, but choosing the optimal one for their data is really difficult, and even if they are chosen, connecting those requires lots of labor and technical expertise. The final problem is really critical. Very often, researchers want to reanalyze their data with different choices of methods and parameters. So they pay a lot of money to get the single-cell RNA data, and they want maximum interpretability. And if they find something interesting, they want to try different settings to maximize their interpretability. But most of the times, it is not really feasible because of time and resource restrictions. Here we present an AI-powered single-cell RNA analysis software as a service. Our service is fully optimized and automated and very easy to use. A very important characteristic is it's really, really fast. For the optimized pipeline to work, even if it is optimized, it takes about at least one full day for the analysis. But our technology reduces the time to 10 minutes and the required memory from 1 terabyte to 20 gigabytes. Because of this really fast time, now we can provide a nearly interactive experience. That is, the researchers can just tune their parameters and try different choices and get the results almost instantly in 10 minutes. Now, you might think it is very insane, because it sounds insane, very unusual, because, well, all the other people in the world take 1 day, even if very smart people, and you guys take just 10 minutes. Well, maybe it's an exaggeration, or maybe they are sacrificing some accuracy, or maybe they are just doing some approximation, maybe 95% accurate, but that is actually not true. So it is very good for me to introduce myself as a professor in Seoul National University at the front, because I can assure you that it is true. That is true because of our technology called Lightning PCA. PCA refers to the principal component analysis, which is a core step in the single-cell RNA analysis. And our new technology is a mathematical technique that can reduce the PCA analysis time by more than 100-fold and memory by more than 50-fold. Since PCA is the main computational bottleneck, using Lightning PCA dramatically reduces the whole analysis time to 10 minutes. And I assure you, there's no loss in accuracy. Mathematically, the solution is equivalent. And our technology is patent pending. One very interesting application of this high speed is the following. Well, because of this high speed, now researchers can get interactive analysis. They can tune their methods and parameters and choices and do the analysis again very fast. But the question is, do they really know the optimal parameter? Do they really know what they want? They can just try and error, but do they really know? So we suggest this. We can, because of this high speed, we can actually run each analysis as a unit. We run the analysis for all more than 1,000 possible combinations of parameters and choices, and let AI select the most interpretable results. It will take more than 1 year with the current solutions, but with our Lightning PCA, it will take 2 to 3 days. This is definitely an area that AI can beat human experts. So again, the researchers pay a lot of money to get the single-cell RNA data, then they want maximum interpretability, and they are willing to pay a little bit more to get the interpretability. And you know, human experts cannot run 1,000 analyses because of the slow speed of the current solution. Here's a comparison table. Well, if the researcher wants to tune the optimization, then they can just hire BI personnel, which is really costly. Another solution is they can just pay for the currently available commercial solutions, but the company takes weeks to months for the initial analysis, and they usually do not allow reanalysis. Even if a reanalysis is allowed in limited times, each reanalysis again takes weeks to 1 month. With our technology, we can give instant results in 10 minutes, and there's a freedom to optimize pipeline, and computational resources not required at the user's end. Reanalysis is unlimited times possible, and each reanalysis takes 10 minutes. And moreover, we can say that we are the only company in the world now that can do this AI exploration, like more than 1,000 combinations and in comparison, to get the maximum interpretability of the expensive single-cell RNA data. Okay, this is our great team. Here's me, and here's Andrew, our CEO. And this is our website, and we are, if you put a JPM 2022, you get $2,000 credit analysis credit for the first one, first 10 personals. Thank you very much, and happy to answer your questions.
Okay, great. Thank you so much for the presentation. Let me turn it over to Noel again of Sopanova Investments to start the questions.
Thanks for the presentation. Curious, how do you define best results, and how do you train an AI to pick, once you run your 1,000 permutations of data analysis, what best results might be? So I'd imagine to train that, you require many data sets with humans actually validating that these are indeed best results.
Oh, that's a really great question. And also, next year we are going to develop the AI feature, and that's really important question that we have to answer. So to answer the question, we have to go back to the what was the initial question. So what are the clinical applications? So there can be several clinical applications. So given the exact cell types, if a specific cell type has a specific large or small expression, does it mean like clinical meaning, like drug response or diagnosis prediction? So those like questions have to be like converted, I think, to the optimal goal of the AI. And the initial step will be how well each clustering will be defined by the clustering algorithm, and what is the differential expression of each one compared to the currently known clusters and known expressions, and then probably at the end, we're going to add pathway interpretation. And I think there are lots of rooms that you have to think about that. Yeah. Thank you very much for the question.
Sorry, one follow-up. Do you imagine expanding beyond single-cell RNA data to others? And how large do you imagine the market is for analytical tools for single-cell RNA-seq?
That's a really good. Thanks for the question. Can I get my PPT again? I'm not seeing. Okay. So thanks for the question. Currently, single-cell RNA has been almost focused on RNA only, but recent technologies now add like openness of DNA and protein again, and methods are really fast developing also. So we're gonna probably next year we're gonna add those multi-omics features to our pipeline so that the researchers with multi-omics can have the most recent and optimized pipeline for that. And again, even if the most recent multi-omics methods that have been published several months ago, they depend on the principal component analysis. So we believe that our fast RNA can be applied to those types of data and make it very, very fast. Thank you.
Great, thank you. I think Aaron No of Intervest in the studio has a question.
Thank you for your presentation. I know that your business model is more like a service provider. And then can you tell us more details about your marketing strategy or business plans about it?
Oh, marketing strategy and business plans. Thank you very much. So we are definitely targeting currently U.S. market. Our CEO is a U.S. citizen, and our company is all foreigners speaking English. And so U.S. market is really large. NIH, you saw the NIH grant, and there are lots of needs there. And other details, actually, we can talk with our CEO. So I'm responsible for the science and technology part here. Thank you very much for the question.
Okay, thank you so much. I think we're just about out of time, but really wanted to thank you for the presentation.
Thank you very much.
Great job on genealogy.
Thanks.
So our next presenting company is ready. That company is a company called GenCore. And GenCore is leveraging its CRISPR system for gene therapy and genome editing. And presenting for GenCore is the CEO, Dr. Young Sam Kim. I'll hand it over to Dr. Kim now to get started.
Thank you for your introductions. I'm Kim with GenCore. Let's get started. Our mission is this: Jesus did miracles, GenCore does routines. Our goal is to develop therapeutically optimal gene editors and to providing best-in-class in vivo gene therapies. Data shows that almost 10 American people are affected by rare disease. And the main cause of the rare disease is a genetic defect. The problem is approved treatments are available only 5% of those people. So we believe that the ultimate solution is to rewrite genetic code, possibly by genome editing technology. And with the progress of CRISPR technology, these dreams coming true. There are huge market because there are more than 500 million people are estimated to be affected. And considering high price policy, as we witnessed at the case of Luxturna and Zolgensma, the TAM size is very, very huge, more than $100 trillion. So we operate several pipelines and the same size as more than $100 billion. And considering the competition between other treatment modality and intra-competition between CRISPR technologies, we expected occupy at least 10% of the same size, accounting for $10 billion. In spite of this potential, there are several hurdles. The first is patent issues. The patent battle is still going on between UC Berkeley and Broad Institute, and other parties are also involved in this combat. The second, safety issues. Cas9 is not safe because most of us have an antibody against Cas9. And there are high level of target issues for Cas9. And third, the delivery issue. The most validated delivery vector system is adeno-associated virus. But the problem is AAV has a limitation in delivery cargo size, 4.7 kilometers. And most of the CRISPR technology has way larger than these limitations. So I'd like to introduce our technology. It's target technology. So instead of using Cas9 or CPF1, we use TMPV as a nuclease. Actually, TMPV is an ancestor protein for all the Cas proteins. And we use an engineered guide RNA, which was published in Nature Biotechnology last year, and we named it augment RNA. So by combining TMPV and engineered RNA, we create a target technology. The best phrase for describing our target technology is self-sufficient minimalist in CRISPR technology. So what's different? First, the durability. So target is so compact that we can deliver our target system in a single AAV. So as we can see, TNPB is just 1/3 of Cas9 protein. So it's very easy to deliver using AAV vectors. The second, the high efficiency. Target outperforms Cas9 and CPF1 in editing efficiency. If I compare editing efficiency with CPF1 and Cas9, target is very, very better in efficiency than CPF1 and Cas9 as well. And third, the persistency. Most of the Cas protein cleaves inside the target sequences, but target cleaves outside the target sequence. It creates multiple events for cleavage that resulting in persistent cleavage. With this property, our system is very, very better in efficacy compared to Edit 101 of Editas Medicine, which is now in clinical trial in the United States. The last property, the one more property is safety. So we use unbiased genome-wide measurement of target activity. Target is more safer than CPF1 and Cas9 as well. And we are safe in terms of immunogenicity because we have no antibody against TMPV because target system originates from archaeon. And the last property is expandability. Most other biotech companies are worried about their large size, but our concern is how to make best use of our extra size. So our values are created by this extra site. So using this extra site, we create base editing system, prime editing system, and other regulators. So a company in the United States is interested in our technology, so we are under licensing negotiation with the company. So recently, we were able to develop adenine-based editing system, which convert A to G sequences, and which is the first platform that can be delivered by AAV, and we secured sufficient base editing efficiency. The last one is target-free. a target fusion alternative tool to prime editing, which was developed by Harvard Medical University. The prime medicine owned the related IP. But compared to the prime editing, target-free system is way better than in terms of efficacy and deliverability and safety. So it is a summary of our advantages in technological terms. So our three platforms, target cut, we can delete or disrupt genes. And when using target ABV, we can substitute one nucleotide in a single base resolution. And target free, to correct or insert or modify in a very free way. So this is our pipelines, 6 pipelines, 2 for ocular disease, LCA10, OSHA2A, 2 for muscular disease, DMD and myotonic disease, 2 for metabolic disease, hypercholesterolemia, and ATTL, which is the same pipeline as Intellia. So I'd like to mention 2 of them. The first one is the treatment of blindness. The LCA10 is caused by the mutation in CEP290 gene. So we used a pair of guide RNA and kill it and delete out of the mutagenic site to recover the subretinal cone cells. And as I mentioned earlier, our system is way better than at 1-1. And second is the treatment of DMD. So we use also a pair of guide RNA to delete exon 51 and recover the dystrophin gene function. And our drugs is so small, we can use the ABP particles, and our platform is way better than any other Cas9-based treatment. So I'm a CEO of GenCore. I was working for the KRIP, a national research institute in Korea, for more than 20 years now. Actually, GenCore is a spin-off company of the KRIP. And we have a very powerful research team, core technology, gene therapy, and plant and animal teams. Actually, GenCore is just a 2-year-old baby, but it's a very early stage. But we've come a long way for the last 2 years. We are wholly dedicated to developing our powerful platforms. And now, in the second phase, we are moving into non-clinical in vivo study. That's why we are going to get funded this time. I'd like to compare GenCore with other leading biotech companies in the United States. Editas, CRISPR Therapeutics, Intellia, Mammoth, Beam, and Prime. So they have huge market cap. But we are small. That means we have much more room for growth. Nonetheless, we have more powerful platforms. Most other companies use Cas9-based platform, but we use very small, compact target system. And we have more powerful pipelines, that is in vivo gene therapies. So we have a research unit, one in Daejeon and another in Seoul, but ultimately we are planning on establishing a corporate in Boston because there are much more good employees, workers, and systems in the hospitals. So that's why we want to get funded this time again. So our aim is we are going to move the IPO pace around 2025. Hopefully, we'll be a leading biotech company in gene editing therapy. Thank you for your time.
Great. Thank you so much, Dr. Kim, for a great presentation. To start off the questions, I wanted to hand it over to Ted Moon, who's a managing director at RM Global Partners. Ted? I think you're on mute, Ted.
Thank you, Josh. It seems like to me that it's a great technology. I have a couple of questions based on your presentation, especially the page 13, the previous one that you circulated. You mentioned that there is a certain percentage for example, 20% to 40% deletion of target gene, and how do you think what level of knockdown deletion are required for clinical benefit? And is it 20% enough or 40% enough? Do you have some I had a chance to review your article as well, but that would be great to hear from you.
Yes, it's great questions. There's no answer about it. The higher the efficacy, the better. But I know there are siRNA-based treatment of Sarepta in the clinical that was approved by FDA. I know their efficacy is just 1%, but it was approved already. So many people argue about that. So I guess at least 10% is enough because 10% makes the people, the crippled people can work and the blinded people can see the light. So, but the higher the efficacy, the better. So we secure the 20 to 40% of deletion efficiency. I guess is the most highest efficiency than other can attain. So. Thank you.
Okay.
Ted, do you have another question? Or should we hand it over to the studio?
If someone else, I'm definitely.
Okay. Yeah. So I think Ji-Woon Chun of KTV Network had a question.
Yeah.
In the studio.
Yeah, thank you very much. Your presentation really impressive, and then thanks to your size, I do believe that there is huge potential. So, but I do think that you are doing AAV nowadays, but recently there is a lot of some researches on the LNP and then other polymer particle to deliver the system like you.
Yes.
So have you ever tried with very direct data is fine, but ever tried about the other some delivery systems?
Yes, good questions. So many people are trying to deliver using LNP or RNP, but the problem is LNP is only targeted to liver tissue, not lung, not brain, or so AAV is advantageous because we can deliver AAV in almost every tissue. So Intellia uses LNP, but the indication is only to limit to the liver because LNP was approved for liver tissue. So that's why every other biotech is trying to use AAV. So, okay, so we can use LNP or RNP because no problem. But we are trying to compare between the two. So if lipid nanoparticle is proven to be better, we can choose lipid LNP. But many scientific data indicate that AAV is very versatile and safe and efficient. So we are trying to use the AAV particle. Thank you.
Great, thank you. I think we're just about out of time, but thank you so much for the presentation, and we look forward to hearing more in the future about your progress. So thank you so much.
Thank you.
So our next presenting company is ready. This company is called InnoPharmaScreen. The company combines proteomics technology with AI-assisted drug discovery. And presenting for InnoPharmaScreen is researcher Sun-Go Seo.
Thank you very much for attending our presentation. And my name is Sun-Go Seo, and I'm a scientist at the R&D division of InnoPharmaScreen. And InnoPharmaScreen is a company specializing in novel drug discovery, especially PPI modulators. I'm sorry, the PDF. Just 1 minute. Really sorry about that. We are experiencing some technical difficulties with the presentation right now.
If everyone can just sit tight for a second, KoreaBIO is troubleshooting the connection.
We're sorry for that. So let me just begin again. So we're in a pharma screen, and we are a company specializing in novel drug discovery. Especially we specialize in the discovery of PPI modulators through a hybrid platform of AI-based virtual screening technology along with our proprietary proteomics platform. So we have a team of very highly skilled and very highly trained team of scientists, management, and external consultants. And we have multiple intellectual properties, especially patents registered in the US. And our business model focuses on licensing out of our assets, preferably at preclinical stages, and open innovation through collaborations and joint research with global pharmaceutical companies. Now I wish to highlight some of our research highlights and some of our major pipelines. As I've said before, our company specializes in the discovery of PPI modulators, protein-protein interaction modulators. And these include PPI enhancers and PPI inhibitors. And we make use of extensive computational methods, for example, molecular dynamics, PPI prediction algorithms, and AI-based molecular design algorithms. And this is combined with our proprietary IPS PPI Explorer technology, which is our proprietary chip technology to validate PPIs with unprecedented speed and accuracy. Our first pipeline is molecular glues, IPS06060. And with these molecular glues, we aim to develop a small molecule that will help adhere our protein of interest with our E3 ubiquitin ligase for ultimate ubiquitination and degradation of our protein of interest. And such modality, molecular glues for targeted protein degradation, is a very novel first-in-class modality. And there are many companies which are working on molecular glues right now, but most of them are at very early stages of development. And there's a very vast avenue for potential research in this field. And what makes IPS06060 molecular glue pipeline so special? The answer is in our discovery strategy. So we have a few highlights in our discovery strategy. There are 4 highlights. The first highlight is our reliable protein-protein interaction PPI prediction algorithm. And with our algorithm, we are able to model protein-protein interactions, which are very complex, with very high accuracy, with unprecedented speed, and that results in extremely high reliability. And the second key point is in the AI-based molecular design algorithm, and using our own unique AI algorithms, we are able to design a molecule which will suitably adhere our protein of interest with the E3 ubiquitin ligase. And thirdly is our molecular dynamics. So we use our own MD protocol, molecular dynamics protocol, for the analysis of the compounds that we have discovered. And by using our protocol, we are aiming to simulate the conditions inside the cell as accurately as possible, and we are met with unprecedented accuracy with this as well. And the compound that we have discovered is then analyzed through the IPS chip PPI Explorer technology for potential binding and ubiquitination. And the details of this technology will be explained in the next slide. So this is a bit more detail about our IPS chip PPI Explorer technology. So we have a proprietary chip technology, and with our own unique assay methods to test for binding and ubiquitination of our protein of interest with the E3 ubiquitin ligase. And with these proprietary and unique assay methods, we are able to immediately verify the computational findings, and we can do this with a very small amount of protein, and the setup and the experiment time is greatly reduced. So it allows for the validation of the in silico methods extremely fast, and also it allows for the feedback into the computational method to train the algorithm and to enhance its effectiveness in the discovery process. And I wish to present some of the results for our molecular glue discovery. So as you can see in the picture represents the molecular dynamic simulation results at elevated temperatures. The gray solid, as you can see in the bottom half of the picture on the the upper left-hand side, represents a molecular glue, potential molecular glue that we have discovered. And you can see that without the ligand, we can see that the protein complex slowly dissociates, but with the molecular glue, the potential molecular glue, we can see that the dissociation of the protein complex is greatly stabilized. And also other data show that our molecular glue candidate is very effective in stabilizing the interaction between our protein of interest and E3 ubiquitin ligase. Our next pipeline is an immune checkpoint inhibitor, especially it's an inhibitor of TIGIT. And it's a small molecule, and what makes this special is that there are no small molecular immune checkpoint inhibitors on the market right now. So it's a very novel and very first-in-class modality, I would say. And with such a small molecule inhibitor of TIGIT, it can be used as a combination therapy with other immune checkpoint inhibitors, such as PD-L1 antibodies, and we expect a great increase in the response rate when used as a combination therapy. The rationale for this discovery is that we wish to develop a small molecule that will inhibit the binding between TIGIT and CD155, which is also called PVR. And we wish to develop a small molecule that's only going to bind to TIGIT, but not to the competitive regulator CD226, which has a very similar structure, and we wish to detect the very subtle differences in the structure. So there are two things. It's a small molecule which has never been done before in the field of immune checkpoint inhibitors, and also with the small molecule we are aiming to secure specificity. And I wish to present some of the key results for the small molecule discovery of TIGIT inhibitors. And as you can see on the left bottom half of the picture, we can see that using our proprietary IPS chip technology, we can see that these two molecules that we have found two molecules using extensive in silico screening and computational methods. We have found two molecules, compound 103 and compound 115, I believe. And these two compounds are very effective in reducing the interaction between TIGIT and PVR according to what we have expected. And also on the right-hand bottom half of the right-hand bottom side of the picture, we see that these two compounds are very effective in inhibiting the interaction between TIGIT and PVR dose dependently, but they do not inhibit the interaction between CD226 to PVR, which is the specificity for TIGIT that we have wanted and that we have intended with developing the small molecular inhibitor of TIGIT. And our next pipeline focuses on the inflammatory response. So we are, the name of the pipeline is IPS07004, and it is an inhibitor of the NLRP3 inflammasome. And the NLRP3 inflammasome is a central, I guess, mediator of the inflammatory response that's involved in many human diseases. So IPS07004, as I said, it's an inhibitor of the NLRP3 inflammasome. And the key importance of the NLRP3 inflammasome is that the NLRP3 inflammasome mediated pathway and the disease is caused. So we are targeting severe asthma and Parkinson's disease as the indications. And targeting NLRP3 inflammasome, it's a very novel MOA that we have discovered. And so that's the key highlight of this pipeline. So it's a small molecule, but it's a repositioned drug, and it's a repositioned drug, and using this drug, and we're using this drug to generate new derivatives, IPS 07004 derivatives for new chemical entities. So I wish to describe some of the key results in using IPS 07004. We can see that in vitro tests show that the treatment of IPS 07004 greatly reduces the release of inflammatory cytokines. And also, we can see that the derivatives are much more effective than the original IPS 07004 itself, as much as 100 times. And most importantly, in vivo, we can say that one very distinguishing factor of IPS 07004 from other drugs is that it's able to reduce the number of neutrophils and eosinophils simultaneously. And also in vivo, in severe asthma-induced mouse models, we can see that IPS 07004 greatly reduces the inflammation biomarkers that leads to the relief of the narrowed airways, which means it's very effective in treating severe asthma in vivo models. So IPS 07004 is a repositioned drug. So although we don't have a new material patent, we are filing a new use patent based on a novel mode of action for our 2 indications. And we have submitted a phase 2A IND for this, and we are performing supplementary experiments, and we are planning to commence the phase 2A trials by the 3rd quarter of this year. And the next indication for IPS 07004 is the Parkinson's disease. It has been recently found that alpha-synuclein is a protein that initiates the inflammatory response in the microglia mediated by the NR-RP3 inflammasome, and that causes the release of inflammatory cytokines that causes the cell death of dopaminergic neurons, which leads to the symptoms of Parkinsonism. And this is a novel MOA that we are targeting for with IPS 07004 that we have discovered. And here are the key results to using IPS 07004 for the treatment of Parkinson's disease. So firstly, we have in vivo results for the MPTP-induced zebrafish models. So we can see that compared to the control, when treated with MPTP, the zebrafish, the motor functions of the zebrafish is completely eliminated. But when treated with IPS 07004, the motor function is restored to near normal levels. And another importance is that we have compared our results with another drug called rasagiline, which is a monoamine oxidase B inhibitor, and which is a marketed drug for Parkinson's disease right now. And it's evident that our drug is shown greater effectiveness compared to rasagiline. And also in microglia, the treatment of IPS 07004 reduces the level of inflammatory cytokines. And also another in vivo results show that in MPTP-treated mouse models, the motor function of these mice is greatly increased with the treatment of IPS 07004, which is explained by the immunohistochemical staining diagram in the bottom right of the picture. We can see that staining for dopaminergic neurons in the brain, we can see that the application of a drug has greatly increased the survival of dopaminergic neurons. And for this indication, we are planning to submit a phase 2A IND. And just to summarize our presentation, we have a very unique AI-based hybrid platform with cutting-edge technology for drug discovery. And with the molecular glues, it's a targeted protein degrader, and it has a very novel modality, and we can target specific oncogenic mutants. Also, we can target undruggable proteins using our MG platform technology. And also for TIGIT, it's a first-in-class novel molecule, so it's an immune checkpoint inhibitor, and IPS 07004 targets very novel mode of action. And these pipelines and the platform that we have allows for continuous development of innovative pipelines well suited for licensing out and potential tech transfers. Thank you very much. If there are any questions, I'll be very happy to answer.
Great. Thank you so much for the presentation. I know we have a couple of questions coming in, but maybe we'll start with Ted, if we could. Ted Moon from RM Global Partners.
Thank you. Yeah, the small company with a lot of interesting technologies development. My first question is how do you prioritize this development? All for licensing out in the preclinical stage, or do you have any certain strategy in just selecting the further development or not?
So basically, we are planning to license out our assets as soon as possible. So for IPS 07004, which is an asthma and Parkinson's asset, it's right now entering phase 2A clinical trials this year. So we are aiming to license out along with the derivatives as a package. And also for our molecular glue and TIGIT, we aim to license out the compounds as soon as we have in vitro and in vivo data on that. Did it
Great.
answer
Let
your
me
question?
turn it over now to Min-Sik Shin from KB Investments, who I think had a question as well.
Yeah, thank you very much for impressive presentations. I just had one question regarding TIGIT pipelines.
Yep.
It's impressive that we're developing a small molecule TIGIT inhibitor. However, there should be a reason why people are developing it in antibody way, noting that not only it blocks this TIGIT, but also it gives ADCC effect, which is the antibody cytokine effects. It's more than like just a blockade. Would this efficacy be feasible for our compound compared to the antibody competitors?
Yes, we are very confident that it will show a very similar efficacy. And with our CHIP results that we have preliminary data on the efficacy of our small compound, and we are expecting with further improvement and further optimization, we are improving, and we expect for the small molecule to have a very good efficacy as well.
Great, and I think Ji-Woong Chung of KTB Network had a question.
Yes, the two questions, the simple question. The one is, how is your approximately the molecular weight of your assets? Because you are doing like some molecular glue, which can give us some more advantage on the size itself. Yeah, that's first question. Second question is that relating to that, and then would like to know your assets, like the PK profile, generally, and then especially for some Parkinson assets, how is the biodistribution data, especially for the brain and plasma ratio or something? And then, so those two questions.
Yep. So first, the molecular weight of our molecular glues. So according to our in silico discovery strategy, the molecules that we have discovered is generally below 450 grams per mole, so Daltons. And for the second question, I'm not a specialist in that area, so I'll make sure to contact you after inquiring specialists for that. Thank you very much.
Okay, I think we do have one final question for this presenter from Inai Park of Daily Partners.
Yes, thank you for your presentation. My question is short. I'm just wondering if you have a medichemist in your company.
Yes, we do.
If so, how many? And since you're making small molecules, do you have your own proprietary chemical libraries? And if so, what's the size?
Yep. So we have a team of scientists who are very special, who are specialists in synthesizing organic compounds. We have approximately 3 people, and we have our own lab for synthesis of our own molecules that we have discovered.
Great. I think we're just about out of time. So thank you so much for the presentation.
Thank you very much.
We look forward to hearing more in future years.
Thank you.
So our next presenting company is a company called PharmCAD. PharmCAD is building computer-assisted drug and vaccine development platforms. And presenting for PharmCAD is Dr. Il-Sang Yu, who is the VP and head of the R&D Center and Assay Development. I'll hand it over to Dr. Yoon, and we'll get started.
Okay, thank you very much, everyone. My name is Il-Sang Yoon. I'm the head of R&D Center at PharmCAD. We are a relatively young company, and we are trying to develop the AI-based and physics-based drugs. So let's see. Okay. Today, in my presentation, I'd like to introduce our manpower, platform technologies, pipelines, and lastly, our business plan. So first, our manpower. We established our PharmCAD, the company, March 2019. We are a relatively young company. However, we already employed over 80 people, and among over 80 people, the 52 of them are R&D scientists, and 35 of them are PhD scientists. Our scientific founder, Dr. Sang-Woo Woo, is a physics professor at Bugyeong National University, and he's an expert in the field of physics-based drug discovery. We are very proud of our manpower. So we have, like I mentioned, 35 PhD scientists. Most of them are chemistry, they are computational chemistry experts and AI and data scientists. However, we also have cellular biology, the scientists like myself, and we have drug developers. Okay, yeah. We built a R&D center in India a year ago, tried to recruit the very highly qualified and the talented the computational chemistry and the data and AI expert there. Currently we have 14 scientists. So they are working on various fields of our drug discovery, including the physics-based drugs and proteins and drug delivery systems. In addition to our Indian office, we also have office in Boston to do the R&D in the future and the business development. From the start, we are focused on open innovation. So accordingly, we have the established R&D collaboration with the CNRS Institute in France for PROTEC and drug delivery system. Also, we have established with collaboration with Carnegie Mellon University for data science and AI field. The strong IP protection is very important for us. So we have registered 3 patents in Korea at the moment. However, we also, filed several others internationally. Next, I'd like to briefly introduce our platform technologies. And first one is what we call Formulator. Formulator is the platform for the design to generate small molecules for protein structure-based, and also we are planning for the RNA structure-based small molecules. So it has 5 functional modalities. So the proteins 3D structure optimization and RNA structure prediction module, and drug generation module, and virtual screening and molecular dynamics simulation module, quantum calculation module, and lastly, toxicity prediction module. We have prepared a very short video for better understanding, so that the next slide will show you the small video, which will explain how our formulator works. Okay. Oh, sorry.
Let's look at the new functions, our updated Formulator version 2.5. Graph neural network technology has been added to generate novel drug candidates. Drug generation, additionally, is trained by specific scaffolds. It can predict and generate structures of novel drug candidates that have more synthetic feasibility, but less toxicity. The candidates by drug generation module are not only structurally unique, but also highly developable. New adopted pharmacophore analysis module and interaction similarities functions have been added to Formulator version 2.5 as well. In consequence, the new version can screen the database about 40 times faster than previous docking methods. In silico high-throughput screening. We have executed quantum calculation for 100,000 data from Kemble database. This database is utilized for improving toxicity prediction, as well as developing AI force field. The more enhanced accuracy of new drug design can be made by our quantum calculated database. The toxicity of new compounds has to be evaluated to make a next progress. The toxicity prediction module of Formulator analyzes HCF, dose, BICEP, CYP, and HERG databases, so it can predict various side effects, such as hepatotoxicity or cardiotoxicity. Based on Formulator's performance above, the related R&D period and cost of in vitro assay can be dramatically saved.
Yeah, that was the brief introduction about our first the platform technology called Formulator. And the next one is what we call FARMBAG. FARMBAG is a simulation technology platform which is aimed to develop the vaccines. So we are developing this platform based on our the accumulating experience with developing the COVID-19 mRNA vaccine together with IGN. So this vaccine platform has two the modules. One is the RNA sequence optimization module, and the second one is drug delivery system optimization module. And using this technology, we can simulate how the RNA and the DDS, such as the LNP, how they interact with each other. So I also prepared a very short, about 2 minutes video as well to better, describe how it works.
FarmVac platform integrates AI-assisted modeling of RNA structure and drug delivery systems in an effective, interactive, and iterative way. It provides optimal solutions to the users and clients to design, analyze, and visualize reliable RNA sequences for any vaccine candidates. Adaptable codon optimizer of mRNA sequences let the researchers find the most stable and highly immunogenic candidate, which maximizes protein expression efficiently in the heterologous systems. For example, FarmCAD can generate the most optimal mRNA sequence, targeting to certain spike proteins of COVID-19 using FarmVac. It means FarmVac can encode the designed synthetic mRNA sequences to discover vaccine candidates. More than RNA sequence optimization, this platform helps to manipulate RNA structure conundrum, as well as prompt pharmaceutically important RNA targeting modules. FarmVac can be utilized to discover or design novel small molecules that interact with RNA. Our FarmVac platform can generate more optimized structure of LNP as one of the significant drug delivery systems. Now, we would like to demonstrate the process of DDS generation and optimization. FarmVac analyzes the interactions between ionizable cationic lipids with the optimal number of lipid molecules, both in the atomistic and coarse-grained scales. FarmVac can simulate how drug delivery system works. Based on this simulation, it provides the best composition of ratio for each of lipid substance. Lipid nanoparticle parameter settings, such as lipid composition, size, shape, density, and pH changes are fine-tunable to provide the most optimized LNPs that meet the user requirements. The DDS module of FarmVac will be the effective platform for the optimal LNPs, mediated delivery of RNA molecules and protein peptide molecules. Therefore, our FarmVac platform makes the vaccine development more reliable and faster.
Yeah, that was our second platform called FarmVac. And now I'd like to briefly introduce our current in-house pipelines. So the most advanced pipeline, as you may have read about it, we are developing COVID-19 mRNA vaccine together with a biotech company in Korea called IGN. And we delivered our mRNA sequence to IGN, and they are taking over, using this sequence and their own lipid nanoparticle, the liposomes, and they are advancing the program to currently clinical phase 1. We also have several early stage, discovery stage molecules for cancer targets, including the AML and the breast cancer and glioblastomas. It's still very early stage. Also, we have a very unique program for anti-coagulation targeting the factor 9A. And in collaboration with a couple of other companies, we are developing CNS targeting molecules for Alzheimer's disease and autism spectrum disorders. Now I'd like to introduce some of our business plan. The way we want to generate revenue is mainly two ways. First one is we want to license out our in-house, the program as soon as possible, hopefully before, the pre-clinical stage. One of the great example would be the mRNA vaccine program. And we also can collaborate with the other companies, to share the burden of financial and R&D burdens. We can collaborate with other companies to develop from start as far as they want to, and then we can collect the future the upfront fees and like a milestone payment and sales the royalties kind of things. So our target customers, the first one would be the global big pharmas, who wants to expand their internal programs. The great example would be just this month, the UK AI company called Excentia had the multi-billion-dollar deal with Sanofi. So hopefully we can emulate or copy similar types of deal in the future. We also targeting the the new companies like who just entered the biotech industry, but they have no pipeline in hands. Then we can give them like early stage pipelines, so we can work together. Also, the NLDO types of company would be our customer as well. Since its inception in 2019, it's been over less than 3 years now. We have accumulated about $20 million in investment money so far. And we are just about to start the next round of investment next month. And if it goes successfully and if other plans are working as we wish, then we plan to do the IPO at the end of this year. Okay, thank you very much for your time and attention. I'm ready for your questions. Thank you.
Great, thank you so much for the presentation. Let's start off with Michael Key on the CBC group.
Great, thank you for your presentation, very interesting. And just to better understand the FarmVac platform as you apply to the COVID-19 vaccine, can you explain how the technology allow you to either increase immunogenicity or what aspect of the COVID-19 vaccine you were able to optimize? And what proof of concept you were able to demonstrate before you went to the clinic?
So I can't reveal all the technical details, but basically our role in the COVID-19 vaccine is the sequence optimization. If you could understand so the codon optimization through the best combination of GC contents and also the best way of having the lowest free energy calculation. So that was their main role of in that specific program. If you are more interested in the details, then we can contact separately, and we will present the details of our technology. But you could say mainly codon optimization and the free energy calculation.
And then you're using iGene's LNP you mentioned. I'm not familiar with their some technology, but I'm assuming it's going to be as good as the Arcuitas and other LNPs that Moderna.
Yeah, iGene's technology is not actually the LNP like any other companies. They have their own proprietary positive ion liposome, so which is which works quite differently from LNP. Instead of spreading, systematically, they only stay where the injection is. Also, they can store that, the final product 2 to 8 degree for a longer period of time instead of cold chain. So that's their advantage.
I think we did have, Michael, we had one question from Ji Wong Chan of KTV Network.
Quite interesting to see those pipelines in the biotech for the anticoagulation, because NOAC is to develop is really, really expensive and costly. And then, so we want to know what is your strategy about NOAC development, or are you going to licensing out early stage, or you want to choose some very smart indications?
So this particular program started from our scientific founder, Dr. Sang Woo's postdoctoral training. So it has been quite a while. In his research group at the University of North Carolina, they thought and still thinking that the factor 9A would be the alternative target instead of the current factor 10. And like you said, there's currently no clinical program going on targeting factor 9. So it's going to be a challenging program. However, at the same time, it's going to be first in class. And our business strategy is not to try going to clinic. So as fast as possible, we'll deliver to someone who can develop into their clinical stage.
Thank you for the questions and for the presentation. I'm afraid we're out of time, but thank you so much for the presentation.
Yeah, thank you very much.
Okay, so we do have now our final presenting company. So our next company is a company called ViroCure, and ViroCure is developing cancer therapies using proprietary oncolytic viruses. Presenting for ViroCure is Head of Strategy and Business Development, Gun Kim.
Hi, I'm Glenn Kim. I'm just Vice President of ViroCure. I'm taking care of some corporate strategy as well as business development matters. I'm just to introduce my company briefly and the technology as well. The ViroCure is the company developing anti-cancer drugs utilizing our own proprietary oncolytic viruses. Current virus platforms are the real virus and the engineered mixomer pox virus. We have our own biosafety level 2 animal lab in our headquarters where we can generate the speedier in vivo data. We have 9 patents registered in Korea, United States, China, and Japan, and 13 patients filed for registration. As you may well know, since the 1918 Nobel Prize in Medicine went to immuno-oncology drugs like PD-1, PD-L1, and the CTLA-4, they are now all the rage in the anti-cancer market and growing very fast as well. The cancer patients who are taking these immuno-oncology drugs reaching more than 50% of the total cancer population. But there are some limits of immuno-oncology drugs as well. For most of the cancer types, the overall response rate is hovering just only 20 to 25%. So they need a proper combination partner to complement the limits and then boost efficacy. The oncolytic virus penetrates the tumor for direct tumor killing and induces tumor antigen exposure. Antigen exposure activates the human immune system, and combined administration of immuno-oncology drugs kills the remaining cancer cells. The oncolytic virus's intrinsic characteristic of oncotropism attracts immuno-oncology pharma's attention. Combination therapy with oncolytic virus and immuno checkpoint inhibitors dramatically increases the response rate and treatment effect for cancer patients. Global pharmaceutical companies continue to buy out the agent or the form even in the preclinical stage, with a single transaction value reaching nearly $1 billion. This is the global anti-cancer drug market with a number of patients and market size and unmet needs. The circle, the red ones are the indications that the BioCare now targeting at. And colorectal cancer and non-small cell lung cancer are the targets the BioCare currently looking at. And these are the largest. These are the largest the patient groups, and then which have less than 5-year survival rate of 15%. Okay, those are the BioCare's current target indication, and with the Rio virus, Rio means respiratory enteric orphan, and utilizing its own mode of action, Rio virus is more meant to cure effectively the cancers in the respiratory and gastrointestinal organs. Bladder cancer and head and neck cancer have a small group of patients, but those are the BioCare's another target indication for breakthrough drug or orphan drug designation. As you can see in this family tree, BioCare cultivated their own platforms using a double-stranded RNA virus, as well as engineering adaptable larger DNA mixomer or pox virus. The most popular viruses currently employed for drugs are reovirus, adenovirus, herpes virus, and mixomer, the pox virus. Our own platform boasts the strengths in cancer treatment efficacy as well as the track record of safety to humans. Those are the most important factors to be borne by anti-cancer agent. Turning to the BioCare's current pipeline, IC402IT is in phase 1B clinical trial in Australia right now. Phase 1A confirmed non-toxic, and then 1B is in progress alone as well as in combination with Keytruda, and with the plans to end up by the third quarter of this year. The currently licensing out has been exploring, especially toward the immune checkpoint inhibitors and the CAR-T, the holders as well. RC402PO is aiming at the first oral administered oncolytic virus anti-cancer drug for treatment of bad refractory MSS-type colorectal cancer with a lower PD-L1 expression rate. Clinical efficacy data were presented at the American Association for Cancer Research last year, showing up a very significant effect in suppressing tumor growth when used in combination with the immune checkpoint inhibitors. MC509N1 is the first in-class novel mixomer virus agent embodying proprietary gene recombination technology. We are exploring and testing target cancers tailored to each patient's biomarkers. Oncolytic virus and NK cell or the CAR-NK cell combination therapy is in development for treating bladder cancer patients with poor prognosis after BCG treatment. This pipeline is joint R&D program with the Samsung Seoul Hospital. RP116M is another reovirus platform. It is patented in the US and capitalizing on its intrinsic properties of reovirus is in development on oral administered colorectal cancer vaccine as the preliminary grant award program from Canadian Cancer Society as well. Looking into briefly the HA BioCure's pipeline mentioned before, IC402IT is designed for maximizing intratumoral effectiveness. It specializes in treating cancers caused by mutations in tumor suppressors like P53, ATM, etc. And it targets to unmet needs of liver metastasized cancers. RC402IT is in the 1B in Australia with the Keytruda and expect to finish up by the third quarter. And then we're going to try to find a possible licensing partner by the timeline. RC402PO is aiming at the first oral oncolytic virus drug for the treatment of colorectal cancer. This is a gut microbiome-friendly agent and expect to induce responsiveness in MSS-type colorectal cancer patient group who suffers a strong resistance to conventional cancer drugs. CT26 is a well-known colorectal cancer that's very refractory to immune checkpoint inhibitors. Triple combination administration of our reovirus, an anti-PD-1 and anti-CTLA-4 significantly increase the tumor suppressive effect by up to 85% and reaches higher effect in complete remission as well. Preclinical development going on faster, and the paper will be published soon. RP116M is a modified form of RC402, where the spike proteins that attach to human antibodies were removed. RP116M is a current backbone of a comprehensive oral administered cancer vaccine for colorectal therapeutics, and this is also the vaccine for the virus vaccine platform on our own, including coronavirus and other viruses which are coming up, possibly coming up in the future. MC509N1 is a multiple transgenes inserted agent which improves tumor lysis function with a synergistic effect of those transgenes. And even when used in combination with the immune checkpoint inhibitors, our data shows most of the tumors killed and close to complete remission in vivo. We hope to license out this agent by next year. And clinical stages, as in the cases shown in the prior slide. Wrapping up this presentation, BioCure visions the global top of 10 viral immunotherapy pioneer that gives good cures for cancer patients. And our goal focuses on the development of a patient or a specific, most innovative cancer drugs utilizing our own oncolytic viruses. Thank you very much for having me.
Great, thank you for the presentation. To start with, I wanted to hand it over to Bosun Hao of Tyburn Capital Management.
Thanks, John. Appreciate the presentation. Oncolytic viruses is an area that I know a little bit about. I used to be a firm that we led the round prior to in BioVex that ultimately ended up selling to Amgen, which is where TVAC comes from. Look, I think my general question, and it's a broad one, but look, I think oncolytic viruses have shown, kind of very favorable, I think, risk benefit in preclinical and clinical studies. But I would say, there's been certainly challenges to kind of make it to mainstream, and I think a lot of it ties to some of the limitations around, kind of delivery, manifesting really this immunity in addition to the cytotoxic or the tumor lysis. And some of that comes back to, ultimately the platform and the limitations around some of these, viruses. And look, one thing I wanted to ask you is really around your reovirus platform, which you're optimizing on. Why do you think this is, highly differentiated? I mean, because we haven't really discussed the differentiation there, and aside from safety, and we all know largely these things are safe. And I think one of the limitations on reovirus too is it's the inability to really engineer this. I mean, it's got this segmented kind of RNA backbone, so there are some limitations there. So could you just speak to why you think the reovirus, your platform, is going to overcome a lot of the challenges we've seen with oncolytic virus approaches generally?
Okay, thank you very much. I agree with you, and certainly the oncolytic virus kind of one of the limitations will be the kind of a delivery issue. So how can we get into the kind of the target, the cancers? And probably, as we are doing in Australia with the reovirus RC402IT, we're just making kind of an intratumoral injection so that we can maximize our efficacy. And in addition to that, responding to the question, reovirus has come a more specific mode of action. Most of the viruses, when we orally consumed, the viruses will be killed when they come through the stomach or the exit. But on the contrary, the reovirus is getting much more stronger when it goes through the gastric, the stomach, and then when it go down to the small and the large intestines. So based on this kind of the MOA, we're just making this reovirus a kind of oral administered drugs so that we can cure the gastrointestinal the organs like the stomach or the colorectal the cancers. So I think some of the it's very not easy, but that's why kind of some differentiation, some innovative differentiation is making at BioCure using this reovirus for its kind of intrinsic characteristics of surviving in the organs when consumed orally. Yeah.
Thank you. We're almost out of time, but I think we do have time for one more question. And I believe Ji-Woon Chun of KTB Network had a question.
But my question is kind of very redundant with some previous questions, so I'd hand it over to Dr. Shim.
I just wanted to check some facts. Your reovirus and myxoma virus both are, it doesn't look like it's a recombinant incompetent virus. It looks like wild type. Is that true?
The reovirus, we have two types. One is a wild type, which is right now a clinical stage in Australia, and the other one is a modified reovirus, SRP116M. This is the virus where the kind of spike proteins are removed. We modified them, and then it'll be kind of, we think that it's very good for intravenous administration as well.
Okay, what about myxoma?
Myxoma virus, myxoma pox virus is very large compared to the reovirus. So we can insert transgenes up to the 5 or 6 transgenes. Right now we are doing the 3 transgenes at the moment, but we're just expanding. If they're going to some negative interactions between among those transgenes, we try to insert more transgenes so they can maximize its efficacy.
Okay, thank you. I'm afraid we're just about out of time. Really wanted to thank you for the presentation, and I wanted to thank all of our presenters today for sharing their stories, and a special thanks to all of our investors, both those in the studio and those who tuned in via Zoom. Really, really great questions. We really appreciate your time. And on behalf of BioCentury, it has been my pleasure to join today's program to help highlight life sciences innovation for Korea. So I'll now hand the program back to Jury of KoreaBIO for her closing remarks.
I need to jump off. So great.
Okay,
good to
thank
see
you,
everyone.
Michael.
Good to see you, Ted. Wilson,
Great
Josh.
seeing you. Thank you so much.
Yeah, nice meeting you.
Thank you all. Really appreciate it.
Thank you all. Yeah, bye.
Well, hi, welcome back. Thank you, Josh, and the 8 companies who have pitched today. Josh, again, I thank you for the smooth emceeing. We found out today that language is certainly not a barrier for the Korean companies anymore. As far as I know, there still aren't many enough cross-border investment going on, but I truly hope that through opportunities like this, our companies seek more investment deals, cross-border investment deals in the future. We'll be back with our global investors right here tomorrow at the very same time tomorrow. Tomorrow we will hear about the behind stories about KoreaBIO and Loonback's licensing out deal and 8 global startups pitching hosted by BioCentury and Sidley Austin right here on Buyer TV. Have a great rest of the day.
Thank you all.
Thank you.
Josh. Thank you for joining, guys. Really appreciate it.
Thank you, Josh.
Great job.
Keeping me waiting. All I want